Okay.
Okay, well, um, welcome everyone to today for this panel discussion on the science, clinical issues, social impacts of COVID-19, the COVID-19 pandemic. I'm Roy Wensley. I'm the Dean of the School of Science. I want to introduce to you the three panelists we have today. And they'll just quickly say hi as I introduce them. And we have Professor Vidya Chandrasekaran from the Department of Biology.
Hi, everyone.
Dr. Carl Buettner, who's an alum from St. Mary's in biology. He's the current CEO and Chief Medical Officer of Durham Bryant. And we have Professor Keith Garrison.
Hi, everybody.
Who's also a professor in the Department of Biology. Stefan Perez was supposed to join us today. He's an alum of St. Mary's who works at the CDC. However, he was deployed on an emergency basis to deal with some COVID responses, and so he was unable to join us today. So I think we've got some questions. Those of you who have registered also sent some questions in, and I think most of the questions I will ask the panelists today will cover what you're interested in. If time permits at the end, we'll have some opportunity for you to ask some questions and we'll go from there. So I'm going to jump right in because we've only got an hour. It'll go fast, I believe. And I'm going to start with Dr. Garrison and just let's start out and ask what are the different types of viruses? What's unique about this particular virus that causes COVID-19, and why do some lead to pandemics and others don't?
Good questions. So SARS-CoV-2 is part of a family of viruses that's called coronaviruses, and they're called that because they have a crown of protein spikes that surround the virus particle, and it's those spike proteins that help the virus to get into the cell. It essentially docks with the cell that it's going to infect. What makes the SARS-CoV-2 virus, or the virus that causes COVID-19, unique is that even though it binds to the same receptor, that spike protein is new. So it binds to the receptor in a new way. And that new version of the spike protein likely came from bats in something that we call a zoonosis, when a virus moves from animals to humans. And because it's a new virus in that sense, the immune system really has no previous record of the virus to draw from, because our immune system remembers previous infections. And I think a lot of our predictions for the next pandemic that we were worried about really centered around flu viruses. And so having the next pandemic come from a coronavirus was a bit of a surprise for people.
So just to follow up quickly, and anyone can answer this about, you just mentioned the flu virus. Well, what's the distinction between this type of virus and something like this new novel coronavirus? How do they work differently, and why are they so different?
Well, different viruses will infect different types of cells in the body. Dr. Chandrasekaran might have some more to talk about in that respect.
Well, also different viruses we have differing degrees of experience with, like flu. Over a lifetime of getting flu and getting vaccinated, we may not have the ability to totally immunologically recognize this season's flu, but we have some. Whereas, that's one of the names of this is the novel coronavirus. It's a virus that the human race has not had experience with. So essentially everybody is susceptible. And, it's the novelty of this virus. And not all viruses are equally virulent to begin with. And this one, perhaps by overly activating the innate immune system, it's more lethal. And so viruses of different types vary in their ability to cause disease related to how and how many cells they destroy and the host response they elicit.
Thanks, Carl and Keith. So Vidya, maybe you can talk about just the basic, how does this affect the body, and what causes these, what's the biology of this virus in the body?
We started talking about how viruses infect the body. So one of the things we know about flu virus, as we were talking about just now, is that flu virus is very specific to the respiratory system. So unlike the flu virus, the SARS-CoV-2 virus can affect other systems in the body, because that spike protein that Dr. Garrison was mentioning that is studied on this virus, that spike protein is the thing that the virus uses to dock onto the cell. The protein on the cell surface that spike protein docks onto is a protein that is known as ACE2, which is angiotensin converting enzyme 2. This enzyme is actually an important enzyme for blood pressure regulation in the body. And these receptors for ACE2 are present in the respiratory tract. So all the way starting from the nose into the trachea, going down the airways into the lungs. They are also present in the cardiac, on the heart. They are present in the GI tract. They are present in the nervous system. So what you start seeing is this virus now has the ability to actually get and infect to all these systems, because it has an entry point in all these places, which is also something that adds to the mortality and the severity of the symptoms that we see with this virus. So one of the first things that happens is, if you think of just the respiratory tract, as we ingest or inhale this virus in the upper respiratory tract, the virus will bind to this ACE2 and then enter into the cell and start causing an inflammatory response. Now, the interesting thing about this virus is that that initial stage, when it actually comes in first in contact with the cells, it does have a job of, it suppresses some part of our innate immunity. And what that does is increase the length of incubation period, right? Because our immune system is not responding to this virus as the virus has gotten and started multiplying already. So by the time our immune system kicks in, this virus is already, you've got a pretty good load, which means the immune reaction is much more severe than just a basic common cold. So now when we talk about a mild symptom of it, right, when people start showing the symptoms somewhere between 2 to 14 days, they have a fever, a cough, that, we all sign off these things that we don't have one of these things to get into our spaces. So we have fever, we have cough. There is, so typically upper respiratory, right? Sometimes a runny nose, sometimes sneezing. But the other things that some people report are things like nausea and vomiting and diarrhea, which is really because of the virus's effects on the nerve, on the GI tract. And then some of the people report muscle pain and fatigue, right? That could be an effect on the nervous system, and we don't fully understand that component. Some people report loss of smell. Right, the loss of smell is because the virus actually kills some of the cells in the nasal epithelium, which affects those and inflames them, which causes that. So you see it affecting many different systems. So that is the mild and moderate. 81% of the people have the mild and moderate. Okay, but then in some people, what happens is about 3-4 days after the onset of basic symptoms, the people start experiencing shortness of breath. They have difficulty breathing. And that is because now the virus, instead of just affecting the upper tract, is now affecting the lower respiratory tract, which is the alveoli, the air sacs, where we actually do gas exchange. And so what ends up happening is that once it infects those cells get inflamed. So they get fluid filled, they start dying. And as they start dying, cell debris builds in, pus builds in. Right. So as some of these alveoli start getting inflamed, that is what leads to the pneumonia that we see in patients. And so you can start noticing pneumonia, and on a chest x-ray, it will look like it's fuzzy, because some of these patients will now start having pneumonia. Now, in the early stages of that, there is no effect on the amount of oxygen that the body receives. So these people still have enough oxygen supply, so they don't need supplemental oxygen. They are classified as moderate in terms of the symptoms. Another couple of days later, depending on the person, it leads to the more severe form. The severe form is where now the inflammation that is happening in the lungs can lead to this release of all the stuff from the immune system that is trying to kind of tamp down the virus. But also in the process, all the stuff that is released is causing inflammation in the blood. Because it's in the bloodstream, it affects all the other organs. And so now the disease becomes more severe. That happens in about 14% of the patients. Those patients end up their oxygen levels drop. They have very labored breathing, their heart rate goes up, their breathing rate goes up, and they need supplemental oxygen. And then when this inflammatory mediators are released in lot, this is called the cytokine storm. You may have heard this terminology. The cytokine storm is where all these inflammatory things that are really released to combat the virus end up damaging the tissues in the neighborhood. And that causes the multiple organ failure that then results in death. And these people need mechanical ventilation because by then the lungs are not functioning. So this is kind of the timeline of how the disease actually progresses in us. Some people, even after they recover, there's a fraction of people that even after 3 months after they test negative for the virus, they still show symptoms. They still have chest pain. They still have difficulty in breathing. They still have muscle aches and fatigue that is still associated with it. And we don't understand why that is, but it can linger on some people.
So what you're saying is our immune system actually can be hurting us.
Yeah. So in the initial stages, it is helping us. And then as it tries to ramp up to counter this threat, that excess activation can actually cause the downfall that happens.
Interesting.
The immune response that's supposed to happen early in infection is not happening until later phases. And because it's happening too late, the virus has already sort of gotten ahead in the race, and the immune response ends up causing more problems at that point in the course of infection.
Well, thanks, Dr. Chandrasekaran. And
there's one term that we might want to define there. She mentioned the innate immune system, and that's not a term that the general public is often familiar with. In reality, there are two big branches of the immune system. There's the innate and the adaptive. The innate is the most primitive in that virtually all life on Earth has some form of an innate immune system, where it's only vertebrates that have the adaptive. And the adaptive is the one that makes antibody and has immunologic memory, whereas the innate is more the first line therapy. And the coronaviruses as a group have evolved a strategy to evade that first line of therapy, the innate immune system. And that's one of the ways it differs from flu and other respiratory viruses.
Thanks. So Dr. Breeden, you're following up on some of this. One of the, in terms of just understanding of, how we know that we have the virus or have had the virus, what are these different types of antibody tests, the viral tests?
Right.
How do they work, and what kind of indications do each of them give us?
There's a lot of confusion over this, and it's really not that difficult. There are basically two major types of tests. One test is seeking to understand whether a person has the virus, an active viral infection, by detecting the presence of the virus, and the other is to detect antibodies to the virus. And it gets confusing because antibodies can also be used to detect the virus. So first of all, are you actively infected? And there are two main ways of doing that. One, detecting the virus, and you could culture the virus, but in this day and age, it's more common to not culture the virus, but to use a thing called PCR, or polymerase chain reaction, to detect viral DNA or viral genetic material. And the viral genetic material is there, you assume the virus is there. And so that's sort of the gold standard for active infection, is the PCR looking for the viral, the COVID, the SARS-CoV-2 genetic material. There's also a rapid test that, the part of a virus that stimulates an immune response is called an antigen. And you can raise antibodies to the virus and use those antibodies to detect the virus. So there are tests for detection of the virus based on using an antibody to identify the presence of the virus. So their viral detection methods are asking the question, does this person have the infection right now? And as with all tests, these tests have false positives and false negatives, and false negatives are more of a problem than the false positives, and they are a bit dependent on the quality of the sample that's acquired. Then the testing your blood for the presence of antibodies to this virus is a way of measuring whether you have had a recent or past infection. It usually takes several weeks for us to mount an antibody response. So if you become symptomatic today, you may not have detectable antibodies in your system yet. So the antibody test, the viral test is good at answering the question, does this person have a viral infection now? The antibody test is good at asking, have you had it in the past? And it's particularly good for what are called seroepidemiologic studies, which a number of those are going on around in California and in the US, where you draw blood from a large number of people, test them for antibodies to get a better handle on the denominator of how many people are infected, since the spectrum of disease with this virus ranges from death to asymptomatic. And so detecting antibodies in the blood will tell you whether you've had previous infection. If you have had that, you may have some degree of immunity, but they're also very valuable for epidemiologic studies to understand the spread, transmission, and how many people have been infected.
Thanks, that's great. I think the two important questions, and we'll get to both of these next, have to do with thinking about, how do we protect ourselves from this? And there's two areas where we have to think about our response medically. For the therapeutics, that is, what can we do to treat people, and the other is the vaccine. So let me just first ask, I'll ask this of Dr. Chandrasekaran. Are there therapeutics? What are they? What therapeutics have been demonstrated to be effective in treating people who actually come down with COVID-19?
The question to, are there therapeutics?
Yeah.
That itself is like an inconclusive answer to that particular question. There are lots of things in trials, lots of things that have been tried. I think that's kind of what we have. This is so new. Some of these have been only tried on COVID patients. They haven't been, and typically they're used. NIH guidelines, NIH has very explicit guidelines on when and what therapeutics are available and when they are recommending the use of it. So pretty much all the ones I'll be talking about are really recommended for use in patients who are in that severe category, where they need supplemental oxygen. Their oxygen levels have dropped enough that they are recommending it. And the benefits in the clinical trials have also been seen only in those patients at mortality rate or recovery rate. You don't see much in the early stages. So there's no convincing data for the mild and the moderate for those drugs. So the one drug that I think all of us have heard about, this is the antiviral. So one strategy that we have is some way to stop the virus itself from replicating and from dividing and multiplying in our cells and causing problems. So the one drug that we've all heard about is Remdesivir. Remdesivir is a antiviral agent. And the way this works is it actually blocks the enzyme that is needed for viruses to divide, to copy their DNA, copy their nucleotides, right? So what it ends up doing is that it ends up blocking the virus from dividing, which then prevents the viral load from increasing and then can block further infection. Okay. There are side effects to it, nausea and constipation, simple side effects. It has been approved for use, or it has been shown to be effective in things like SARS and MERS, so other coronaviruses. It has been effective. In SARS-CoV-2 trials, what they found is that Remdesivir does a good job in decreasing the recovery time, hospitalization time for the critically ill people, where it drops that time from, say, 15 days to 11 days or 18 days to 11 days. So there is a quicker recovery that they can see in those patients, but there is not much data for the early stages. The second thing that people have tried are things, again, we may have heard of it, is like convalescent plasma, where you can take the plasma from people who have contracted the virus. Some of these tests, again, sometimes they've looked for whether they had antibodies, other times they haven't looked at the antibody titer, so it's very hard to know if that makes a difference. If the convalescent plasma is really effective. But that's another way, is that they take the plasma from the people who have been infected, and then they go in and they inject it. Again, this is for critically ill, because again, plasma comes with its own risks. So they are not giving it to the mild and the moderate. The third thing that people think about are steroids, like dexamethasone. That is another one that has been, that is being used. Again, it's again given to the critically ill, the people who have had a drop in oxygen levels. And what a steroid does, what dexamethasone would do, is to actually suppress the inflammatory response. So it ends up being an anti-inflammatory. So this is really effective in that last stage where the immune system is going crazy and putting out so many of these inflammatory things that increases the damage. Something like dexamethasone can suppress that. Mm. There are a few things that are in trials, like the monoclonal antibody cocktail, the ones that are specific to SARS-CoV-2. There are some antibodies that are in trials. There are also trials in process about some HIV drugs that are being repurposed to see if they would work with SARS-CoV-2, antiviral HIV drugs. And then there are some antagonists, some blockers, some things that can block the inflammatory response in other ways by binding to some of these molecules. Those are also underway in testing in clinical trials. So none of those are approved for use, patient use as yet.
So there's not a lot of hard evidence for any particular therapeutic, it sounds like that.
No. The trials are, there are just few trials with small populations, and most of them have been done in hospitalized patients and the critical ill, severe to critically ill category.
Well, I mean, currently there are over 500 clinical trials going on here. You can think of therapy broken into several different buckets. One is to stop viral replication. Remember, viruses replicate by taking over our machinery to make their progeny. In that, that's how they replicate. And then there's a immunologic approach where you're trying to do passive immunity, give somebody immunity. And then there's general supportive care, and that's the use of oxygen and ventilators and extracorporeal oxygenation. And that would be called, sort of general medical care and supportive care. And that has improved greatly. The case fatality rate has fallen off because the docs in the trenches have learned better how to manage these patients, how to recognize, to check their oxygen levels sooner and intervene sooner. In general, I would agree most of the publicity with the antivirals have been with late stage patients, but in general, if you look at antiviral therapy, highly effective antiviral therapy, consistently only works if you start early. Viruses do not divide. Viruses grow by one-step growth curve. By that, I mean, with bacteria, one bacteria become two, two become four. Whereas with viruses, one virus infects the cell and then releases thousands of other viruses all at once. So earlier you intervene in that replication, more of an effect you're going to have. So historically, antiviral therapy, the effective antiviral therapy we have has only been effective when given early, not late. Late in the disease, the patient's problem with COVID is less about the viral replication than it is the response of the body to the virus that's there and to the tissue damage that has been done. So it's challenging.
And I can
antibody is essentially a man-made convalescent plasma. So they get yeast or bacteria to make the antibodies that you'd want to have in convalescent plasma. And it has the advantage of it can have antibodies that are very effective and at known quantities, and you don't have the risk of bloodborne pathogens with the monoclonal antibodies that you have with convalescent plasma. And they use a cocktail of these monoclonal antibodies because they can have antibodies with different affinities and hit different parts of the virus.
And I can see how it's counterintuitive to use a immunomodulatory therapy like corticosteroids to try to bring down the immune response in the setting of a virus infection. It would seem like you'd always want to be boosting the immune response,
let's be clear.
but it reaches a point where the immune response becomes your biggest problem.
Yeah, but it's less to be immunosuppressive than it is to be anti-inflammatory. What they need to turn down is the inflammation that's causing destruction.
So now I think one of the thanks for those thinking about the therapeutics, which we hear about, I know all the time. The big question here is about the vaccine, right? Everyone is awaiting a vaccine. Lots of people are still not too sure they'd even take it if it came out. But I just want to understand, how do vaccines work? And in particular for the virus that causes the COVID-19, how does it work compared to other kinds of vaccines that have been developed? In my head, my question is, I know we have vaccines for like the measles, which you get once in your lifetime and it works. We have certain other viruses that you can't really get a vaccine for because it just mutates constantly, and so it can't keep up. So what's the likelihood of something developing here? Dr. Garrison, I'll just have you start with talking to the vaccine question.
Our typical vaccine approach is to give pieces of the virus to people in order to allow that adaptive part of the immune system that Dr. Buettner was talking about to kind of have practice at responding to the virus. It's kind of like a fire drill for the immune system. And once the immune system knows what to do, it develops memory. And then having that memory response is a lot faster and a lot more efficient. So that's the typical approach to developing a vaccine. So like that's the way that the measles virus works, or the measles vaccine. That's the way that the polio vaccine that's used in the US works. And the ability to give a vaccine like that and provide lifelong immunity, that sort of depends on the vaccine. Polio and measles appears to give lifelong immunity, whereas influenza vaccination, you need a new vaccination every year, and that's because of changes in the virus that occur every year, as well as waning immunity as you get further and further away from the vaccination. In the case of the SARS-CoV-2 virus, the vaccine candidates that are furthest along in the process use a different technology where they're delivering RNA from the virus in order to give instructions to your cells to produce pieces of the virus and then provoke an immune response that way. And this is a different way of approaching vaccination.
Roy, to answer your question, it's a little bit, not all viruses are equally immunogenic. Not all viruses are genetically stable. And so, the great successes we've had, mumps, measles, rubella, chickenpox, those are, very immunogenic. The vaccines stimulate very good immune responses, and the viruses have remained genetically stable. With the flu vaccine, is constantly, changing it to the way it looks to the immune system. So we need different vaccines. So, there's still, great hope for in general, the viruses can be classified by the type of genetic material they have as DNA viruses and RNA viruses. And many of our successes have been more with the DNA viruses, and these, especially respiratory RNA viruses, have been more challenging.
In terms of the typical timeline, I know everyone's very concerned about this in particular, which in the United States, we haven't quite been as, eager to get a vaccine developed in recent history. What is the typical timeline for vaccine development, And are we getting better at it? Maybe is a question I might ask in terms
Well,
of.
I mean, it's pretty, I've been involved in a number of vaccine development efforts, and it's pretty straightforward. You break it down into the parts. The first part are immunogenicity studies and safety studies, where you make a vaccine and give it to people to first peek at safety and to see what level of immune responses you get.
Mhm.
But then the big the challenges are the efficacy trials. Think about with drugs, you take sick people and give them a drug and see if they get better. With vaccine trials, you have to give it to healthy people and wait to see if they acquire the infection. And so it's time consuming there. And to some extent, how long it takes is dependent on the rate of infection in your study population, as well as the efficacy of the vaccine. And I think how to do vaccines, we know how to do that well. And I think the current COVID vaccine programs are going on. And the big thing giving them a boost is usually commercial companies developing vaccines don't manufacture it until they know they have a product. And what the government has done is say, we'll buy it from you. And, which has enabled them to fund the manufacturing of it. And manufacturing these vaccines are tricky for a number of technical details, like, they have to be able to make it 3 times in a row and other technical things. So, I think there's every reason to believe the production, the development and production of a COVID vaccine is moving along quite well. What I have a bigger concern about is everybody thinks this will be over once there's a vaccine. And I think you need to think about there's vaccines and then there are antivirals. A vaccine is aimed at decreasing the number of people infected and perhaps even the severity of the disease by lessening the viral burden of those that are infected. But the antiviral therapy is aimed at more preventing people from dying. And we actually need both a vaccine and we need effective, safe therapies if we're going to promptly control this. As illustrated by HIV, we still don't have a vaccine for HIV, but at least in the developed world with people with access to healthcare, it's well cared for based on antiviral therapy. So it isn't, do we need a vaccine or do we need an antiviral drug? The answer is we need both. One to decrease number of people infected and the other to decrease number of people that are dying. And if we could greatly impact the mortality rate, all kinds of things open up.
So I actually was sent a question here from someone who's watching. Michael Crockett is asking. This is a kind of a deep technical question, but this has to do with the cytokine storm that a couple of you have already mentioned. He refers to there is an approved anti-cytokine therapy, I guess. He calls it MAB. I'm not sure what that is.
Monoclonal antibody.
And okay, it says it doesn't appear to have an effect in dampening or improving patients who are showing severe symptoms, and he's just wondering what's the explanation.
So I've been looking at some of those data that came in from some of these monoclonal antibody trials, and one of the things is these trials are done on very small populations,
right?
And with it depends on the severity of the disease as well. And there wasn't anything, I don't think they looked at whether the particular cytokine was upregulated and how much it was upregulated and what was it as a fraction of that big cytokine storm effect that we're seeing. So the data are not robust enough for us to make a call. And I think Genentech Roche is trying for another round of studies that is more controlled with a larger population to address whether these antibodies actually work or not work. I think essentially the jury is out on it. There is something that says there's a minimal effects, another study that says there is no effect.
So
stages, kind of how I see it. There's still more going
on. In just thinking about our future and what's going to, how we're going to be able to, live in some sense of normalcy again. I wanted to look at some questions having to do with that, but So maybe I'll ask first, like, just your opinions of what would be better to have? And I know both are obviously having both of these things will be great, but is it going to be more effective to have a highly effective therapeutic, a highly effective treatment, or would a good vaccine be our best solution? Which
We
of those?
need both. There's not one thing that's going to solve this. And
Yeah.
we need both of those. We need something to decrease the number of people infected and then decrease the mortality rate of those that are infected. So it's not either or, the answer is both, because they do very different things.
Go ahead.
And if I had to pick between the two, I would actually pick the third one, which is masks, social distancing, better behavior, because neither one of these, even with the vaccine, right? The access, as Dr. Buettner is pointing out, all of us getting it is going to be an issue. And when do we get it? And how long that immunity lasts. So some part of it is going to be that how we approach the problem. And when we talk about the severity of the disease, I mean, most of 81% of the people end up in the mild and moderate category. So, so the therapeutics is really helpful for when people progress. That seems to be where we are, but that early management is more beneficial.
But a highly effective, safe, and if it was oral antiviral, even those that with mild disease could take it and decrease their period of infectivity. And I don't disagree that, as the head of the CDC said, masks are probably more effective than the vaccine, but how they became a political statement is one of the many mysteries of this vaccine, of this epidemic.
And I think that's one of the lessons that we've learned from HIV is that by getting viral loads down to very, very low levels, you can not only manage the disease, but you can also limit transmission. And I think that's really important.
I mean, I think that's what, in terms of getting back to some sense of normalcy, that's where we're wondering, right? How do we minimize that, the transmission, right? And so in that vein, maybe Dr. Chandrasekaran, you can talk to the point of what are all the known risk factors for number one, getting the virus, first of all, and then developing the COVID-19 response, and then what are some ways to minimize those?
One of the things we know about this virus, and we've all heard this, is that it affects disproportionately or has severe disease in people that are over 65 years old. So for example, if you look at the data on CDC, what it says is that people over 65 years account for about 15% of the cases in the US. But in terms of fatality and mortality and severe disease, they account for 80% of the mortality that is associated with the disease. So we clearly realize that age is a factor. And age ends up becoming a factor partly because, a lot of this virus's effects, the inflammatory response that we see are because, you see these blood vessels becoming a little more permeable, so there is leakage. And so some of that those changes are already happening in us as we age. So having the virus in there kind of pushes a process that's already ongoing. So that is one. The other things that we see that seem to associate, when we look at the mortality rates and the severity of the disease, people call it comorbidities. What you see there are things like diabetes, hypertension, other pulmonary diseases seem to be associated with higher risk of severe disease. Mm-hmm. And obesity is another one. So in some ways, some of these other factors, probably because the immune system is already somewhat activated in these patients, there is some vascular problems already. Those tend to push the patients more quicker to going from a mild, moderate to a severe condition.
In terms of thinking about the risk factors not associated necessarily with an individual's health, but with just, how we spend time with each other, how we interact with each other. What are some of the more I guess I'll call more risky things to be doing in terms of transmission.
Well, that's easy. Not wearing a mask and not social distancing. It's a quite proven. It's been. It's absolutely well known. That's why the outbreaks that are happening stemming from social and family gatherings where people are not wearing masks and social distancing.
And the long incubation period, the 2 to 14 days, and the fact that a lot of people are asymptomatic.
Mm-hmm.
Right? That combination makes it really difficult to know if I am infected or not, which is why we need to limit some of these other risk factors.
Yeah, but even those that become symptomatic, they're the 3 days or so, more or less, that before they become symptomatic when they're highly infectious. So even if everybody infected became symptomatic, that window from infection until the symptoms begin, they're highly infectious. And so you have that with a whole assortment of viruses.
Is that, I mean, it sounds like that's a typical thing with a virus. Those of us who don't feel sick can still be contagious, is what you're saying.
Yeah, I mean, there's a certain level of replication that makes you infectious, but there needs to also be a certain level of tissue destruction before you become symptomatic. And there's that spot there where it's replicated enough to be contagious, but not yet enough to create symptoms.
I also know that thinking about this, the social impacts of this, that communities of color are being impacted at much greater rates than other communities. What are some of the reasons for that? What can be done?
I think there are two things going on here. One, it's the social injustice thing, actually a greater prevalence of comorbidities and less access to healthcare. But with any virus, virtually any virus, you infect an outbred population, there's a spectrum of disease, and that spectrum of disease can be influenced by yet unknown host factors. For example, there's a couple of compelling studies that say type O blood type is less likely to get that disease than type A and B. And it isn't that your blood type necessarily causes that, but it's a genetic marker, and that people of different genetic backgrounds will have, could have different susceptibilities to different, to disease severity. So one, there's the natural variation in the spectrum of disease in an outbred population, but unfortunately, there are also the social issues of increased comorbidities and lack of healthcare access in some segments of our population.
So if you look on the CDC website again, and there is a COVID race data tracker, which is also a very useful tool to try and look at the socioeconomic pieces and how much they influence the prevalence of this disease in certain population. And what the CDC data shows is that African Americans, Hispanic, Latinx people, and American Indians, Alaska Natives are almost 3 times more likely to be infected with COVID compared to the white population. And the hospitalization rate, which is stunning, is that they are almost 5 times as likely to end up in the more severe category and require hospitalization compared to the white population. Interestingly, some of the death rate numbers come closer to the white population. The African Americans still are about twice as likely to die from coronavirus as opposed to the white population. So there is data, and there have been a few studies of late kind of looking at what is the underlying cause. Is it genetics? Is it environmental? What is it that gives this sort of differential, the disparity that we see? There was one study which is very new. I think it was published in August or so. It's done in Louisiana in their healthcare system, which has about 30% Black people as part of their health network and 70% Caucasians. And what ended up happening is that 70% of their COVID cases were African Americans, and 70%, around 70% of the deaths were African American. And again, what they came up with is there were two things. Some of the clinical markers that were being presented in the African American population were different, in terms of which ones were coming up higher, was different from what you see in a white population, suggesting that there might be a genetic hyperactivate, something in the immune system that might be different. But a lot of things they found was that there were lots of comorbidities. Many of these African Americans lived in low income conditions, poor nutrition, being essential workers, so had to be out there working with lots of people. They also had to live in crowded spaces. And all of those could be other aspect, as Dr. Buettner mentioned, that needs to be looked at to really fully understand the spectrum of the disparity. And another thing that came up for the Latinx population is the lack of information, right? Or there needs to be better information dissemination to help with managing that early stages. So the people weren't going to the hospital or weren't managing it in those early stages, which then led to more hospitalization at the later time. So,
and these are all reasons that the vaccine studies are working very hard to recruit a diverse population, because it's very important to understand efficacy across the broad diversity of our population.
Well, yeah, and the logistical thing is, ideally you do the vaccine at the highest risk of acquisition for a whole variety of reasons.
Right.
You do it in a very low attack rate population, it takes longer.
So I'm gonna ask at least one more question, but I also want to know those who are watching us. You can submit some questions, and we'll have a few minutes here toward the end to answer any of the particular questions you might have. Just so you know, I mean, we can't give specific advice about county, decisions, state government decisions being made, specific medical advice, things like that. But anything following up on some of the things we've been talking about would be great. I mean, I think the $1 million question, and I know I'm asking this is speculation for all of us to think about. I really appreciate, first of all, hearing from the three of you, because you really have the scientific knowledge of how these things work and what's being done to combat it. But I mean, just thinking about how do we approach opening our society up, right? I mean, we're already, I mean, I know government plans have been developed to look at how we begin to open up particular businesses, particular activities. And I'm going to admit my selfishness. I'm most interested in, like, how do we begin to open up classrooms and things like that again? What kinds of minimal things are needed before that becomes a safe activity?
Well, I mean, everybody wants to know what they can do just to protect their little part of the world. The biggest driver of it, what's the overall, rolling 7-day average of number of new cases? Less there is in the US, the less there is in California, the less there is in Contra Costa County, and the less there is in Moraga, easier it is to do things. So the number one driver of this is the rate, the 7-day average of new cases. How much is of, how many active cases of disease are in the community. And first and foremost, that number needs to be down. Yeah. And then once you get that number down, then, all the things they're doing on campus now, which have, last I heard, proved they work, of doing, surveillance testing, making sure people, if they're sick, self-quarantine, and availability of testing and wearing masks and social distancing, that will enable it. But if all that stuff on a background of a low 7-day average new cases will work. Yeah. All that other stuff in a small part of the world, with the larger part of the world ever increasing number of cases, it's not going to be as effective. So, I mean, pandemics are part of the human experience. There was a little anomaly in human history from the time polio vaccine was widely used in the industrialized world until HIV came on scene. There was a small period of time there where otherwise healthy young people didn't die of infectious diseases. The reality is that's an anomaly, and that epidemics and pandemics have been part of our experience, and we know how to address them. And what needs to be done is the 7-day average of new cases needs to be driven down, and that will allow people to open up and be safe again. It's not that hard.
I also think, I think we will also, each of us will have to take personal responsibility, because to some extent, right, for us to be able to open up St. Mary's and get classes in, we have to be cognizant of social distancing and be cognizant of the cleaning and things in our own spaces, right? So even if the cases are just low, not high, and we can open up, right? Because we can see the roaring back of the pandemic in Europe, right? And that is really, again, something for us to be mindful of, is how, if we open it, what responsibility do I take? It means the state has released the responsibility, but it then falls on me to make sure that I am making sure I'm taking all the steps to keep it safe for me and for everybody around me.
Yeah, it does. Oh, go ahead, Keith.
Oh, I was just going to say that I think continuing to work on testing methodologies that are both rapid and accurate, because generally it seems to be sort of a trade-off between those two factors. So I think being able to do both, continuing to work on developing tests that may be capable of both would be very beneficial too.
Do any of you know what is, I mean, I know that there's the two tests, I think Dr. Buettner mentioned the rapid test and then the PCR test for looking for active infection. What is the rate of false results for those tests?
Well, there aren't just two tests. There are multiple ones in each category, and it ranges dramatically. And I don't have that at my fingertips, but some are better than others. Some are given emergency approval, emergency use authorization without being thoroughly vetted, and the actual numbers of false positive and false negative rates in some of these tests may not even be widely known. So, the push to get testing out there resulted in not knowing as much about the performance of these tests as we might like.
And those PCR-based tests are able to pick up low levels of infection. They're very sensitive, but they take longer. Whereas those more rapid tests that are picking up the viral antigens will be faster, but it's going to take you a while, as Dr. Buettner said, to reach that point in the infection where you're producing enough antigen to be able to be picked up by those tests.
Well, qPCR can be done pretty quick, but there's actually a limitation on the amount of equipment and how many, the hardware and reagents, limit it too. The actual test, easily done overnight, but not within minutes, like the very rapid one. But overnight would still be actionable, and at a price where it can be done more widely. $75 to $150 a pop, limits its use and access.
Yeah, cost really is the third part of it. Speed, accuracy, and cost really would be.
I know that we're spending quite a bit of money this year on testing our students on campus. It's not cheap.
Lack of the general public understanding that all tests have false positives and false negatives, and none of them are perfect. And somehow people have a hard time accepting that.
We want certainty, don't we?
Yeah.
I think another thing to overlay on that is that with those antibody tests, their ability to accurately predict a positive for the antibody test in terms of, when the test says you have antibodies, do you really have antibodies, is dependent on how much the disease has already circulated in the population.
Hmm.
The tests essentially get better as the percentage of individuals in the population gets higher. When it's still really low or relatively low, the probability of error gets really high unless you have a super, super, super accurate test.
Someone on the who's
Right.
you asked a question, which this is more for I think Dr. Chandrasekaran and Dr. Garrison. Is it possible to get some of our science students involved in some kind of COVID studies or responses? I mean, I don't think I mean having them get the disease, but thinking about what they can do in terms of helping understand this pandemic better and contributing to it, to the solutions, I'll say.
Well, I know this is one of those, well, I was going to say once in a lifetime events, but I don't know whether this is a once in a lifetime event, but I know certainly it's going to change my research program. A lot of the work that I've been doing has been involved in studying corals as a model system to understand that ancient innate immunity. And I think understanding that it's some of those processes that go wrong early on in SARS-CoV-2 infection that can lead to severe COVID-19 disease. I'd really like to start exploring that more in the future when we can get back to campus and lab research.
There's lots of resources that are more computation based, which are definitely something like at an undergraduate institution. You know, one of the reasons we had thought about trying to get into this, could we develop something with SARS-CoV-2 testing or help with that testing when UC Berkeley started that initiative. But the problem is this is a virus that is transmitted through the air. So working with it gets tricky, so we don't expose our students to something. And so something like a computational approach where we can look at some of these proteins, because there's a lot of data, and start thinking about where potential drugs might bind and look through databases would be a great way to further the research and not get them in risk zones.
So we just have a couple of minutes. I don't know if there's any last 30-second thoughts anyone would like to contribute, but we're coming to the end of our hour. Well, let me just thank Professor Chandrasekaran, Professor Garrison, and Dr. Buettner for joining us to educate us more about this virus and the effects that it's having on us, and really appreciate the time that they spent. And I appreciate all of you who are here to listen to the discussion. I think it'll be archived, so you can always go back and watch it again if you want. So again, thank you very much, everyone.
Thank you for hosting it and moderating it, Roy.
Yeah.
Thank you.
Thanks for being here.